Okay, welcome. This is going to be Lab 3 involving tissues, basically histology, and the integumentary system. Exercises are focused on the Syracuse lab text 5.1 to 5.5 and also exercise 6. When you do the filling out of the handout sheets, you will need to complete through 5.1 to 5.5. We'll do the subsequent sections when we get to the muscular and to the nervous system. So let's get started. The objectives are to review the exercises in 5.1 to 5.5 and use the microscope to review tissues and tissue slides. If you don't have the capability of doing that, such as it's an online course, no problem. Review the sheets for each of the exercises, then review the videos. I've selected videos that put emphasis on making discernments of what the structures are at the microscopic level, and you'll see actual tissue slides, and they'll help you to understand the structure as well as the function. Second, to review exercise 6, which is the integumentary system. There are going to be some microscopic slides from the perspective of images in the videos also about the structure of the integumentary system. You do need to review, take the worksheet or handout sheet as it's called, and complete that at the end of exercise 6. As always, I have uploaded these and set them up in a PDF format. You will do the assignments, filling in the certain areas, and then upload this. This must be done by Friday of this week by 5 p.m. First off, we're going to go through the epithelial tissue. This is classified by two structural criteria. Number one, the number of cell layers. You either have simple, which is just basically one cell layer, or stratified, which is multiple cell layers. Then we have to deal with the shape of the cell. Squamous is kind of flattened, somewhat tile-like. In essence, very little volume for cytoplasm, centrally located nucleus. Cuboidal, the shape is somewhat cube-like, and therefore you're going to have, again, a centrally located nucleus with more cytoplasmic volume. Columnar is going to look like a column or a rectangular shaped structure. The nucleus may be a little bit more oval in shape, but you note that you will have a much larger amount of cytoplasm. Keep in mind that that's room for organelles to be doing certain biochemical functions. Here's an example here in the diagrams of squamous, cuboidal, columnar. Notice that in columnar, the nucleus is somewhat oval shaped, almost elongated to fit into the space of the column. This is under simple epithelia. Under stratified epithelia, you're going to notice that you have squamous, and what happens is as they begin to move upward, they're produced at the lowest part here. This is the basal area where you have cell division mitosis. As you get up to the upper layers, these cells flatten out. They become very squamous, and they're also there for a protective layer, and the cells can be shed off, but don't worry. These cells down here will eventually move upward and replace those cells that are sloughed off. Cuboidal, you don't see many layers, but you do see some. Again, you're going to have a centrally located nucleus, and you may see this as being somewhat central and somewhat more or less spherical. Columnar. When you have stratified columnar, a lot of times what you're going to find is the first few layers closest to the area where you're having what they would call the basal area, the cell of first development. They're somewhat more irregular or cuboidal in shape, but then as they approach the uppermost area where they're facing some type of lumen, in other words, a lumen is a term for space, you're going to see that they fully assume or fully take on the more columnar shape. Here's another diagram showing you always epithelial cells will have a basement membrane underneath. Okay, this is to provide support and contact for the cells. In some cases, the basement membrane may be sophisticated, as we will get into this when we get into histology of tissues. So where do we see these cells? Simple squamous is in the inner lining of the blood vessels. That's where we call these endothelium. Okay, for example, capillaries are basically just endothelial cells. The lining of body cavities, such as the mesothelium. Stratified squamous are found in the inner lining of the esophagus, the inner lining of the vaginal canal. Keratinized stratified squamous, this is where you're going to see in the epidermis of the skin. Now, keratinized means that there's a lot more of a particularly tough water-resistant protein called keratin present. Here you can see in the skin, this would be up these upper layers, the epidermis, and they are going to be loaded with stratified squamous keratinized epithelium. Here you see some of the linings of the peritoneum and the mesothelium, and again, simple squamous epithelium. Now, when we get to simple cuboidal, one of the interesting things that you're going to find with cuboidal is they usually function structurally as sort of like the formation of tubes or tubules, okay, or ductwork, DUCT. So the kidneys, the renal tubules, will see very simple cuboidal. They'll be just the cubes in sort of a circular arrangement. Also, you'll see these in liver cells. Stratified cuboidal will be in the ducts of the sweat glands and the mammary glands. Transitional. Now, one thing with transitional is, keep this in mind, it changes in shape depending on the pressure that is put upon that layer. So you'll find this in the inner lining of the urinary tract. This includes the urinary bladder, ureter, and part of the urethra. Think about the urinary bladder just for 1 second. As it fills up with urine, it kind of stretches the layers of cells, which appear more spherical. But as they get stretched out, as this containment wall or containment bladder gets stretched, the cells take on more thinner and thinner appearance in some regards. Let's take a look at some of these diagrams here. Here you see a kidney tubule. Notice that they're all nicely cuboidal, simple cuboidal. And you can see how this arrangement is. If you go to the next slide, we see the sweat glands. Here you're going to have stratified layers, stratified cuboidal. The lumen is the central point where the liquids are moving through, okay? But this is what you would normally see with a sweat gland duct. Here we have the transitional cell lines. And as you can see, these are nice and round here with an empty bladder. But when you start having the pressure as the urine is filling up in the bladder, the upper layers kind of thin out. It's almost as if you're stretching them out. And as you stretch them out, the layers are still somewhat there, but the shape has changed. Now, this is sometimes harder to get in a microscopic slide because unless you freeze them instantaneously when they're stretched out, it's kind of difficult, but you can definitely tell the difference because of the fact that they are rounded, and you'll see there's sort of a non-set arrangement. As always, with all epithelium, you're going to have a basement membrane there, okay? Simple columnar we'll find in the inner lining of the gastrointestinal tract, such as stomach, small and large intestine, okay? Pseudostratified columnar. Pseudo means false, so false stratification. How does that work? Well, it's almost as if you were looking through an arrangement of trees. You see some that are tall with their tips, others that are shorter. But that, if you look on this from a microscopic slide, gives you a false image, a false appearance of stratification when it's not. You will see pseudostratified columnar in inner lining of the trachea and large airways of the lung. You will have some cases of stratified columnar, but this is rare. You see this in the large ducts of the salivary glands and the pancreas. Here's an example of the intestinal lining. Here you have simple columnar. Notice that you have a specialized structure at the very top here, the microvilli. Notice that the nucleus is somewhat stretched out, more oval than spherical. And it has a nice basement membrane underneath here, consisting of some connective tissue, etc. So you're going to see this in the intestinal lining, but then you're going to go over to here. Now, this is where it's going to get a little bit tricky, but you should be able to pick it up pretty quickly. Look at the upper adaptation here. This is cilia. These are the pseudostratified, and they call it columnar epithelium. They look column-like, but you know what? This is not sitting on top of this cell. This cell is stretched just a little bit, and the rest of this cell is still attached to the bottom. Remember how I said with all the columnar cells, you'll always have them start off somewhat round-like, but then eventually they start growing and expanding and eventually become columns. How can you tell pseudostratified from basic stratified columnar? Well, one thing is this. You're going to see irregular patterns here, not a set of a layer, then another layer, then another layer, then another layer. You will also see how the cytoplasm and the nuclei aren't always nice and even in their distribution. But finally, one of the other things that'll really help is the fact that you're going to have cilia here. Not microvilli. Microvilli are for absorbing, increasing the absorptive surface area. The cilia here move, they beat. And this is where you're going to find them very commonly in the trachea, because as you have this thin layer of mucus here, which picks up bacteria and dust particles, etc., the cilia are all beating this to move this material, we'll say just basic contaminants in the air, upwards to be eventually either swallowed or spit out. Okay? Now, the other thing you're going to find very common in trachea tissue, right next to the pseudostratified, is single cells, such as you see here, and these are called goblet cells. A goblet cell basically has an attachment down to the basement membrane, but it's involved with making a lot of mucin, which when mixed with water becomes mucus. These single cells produce a significant amount of mucus that helps coat these layers so that you can basically remove different particles, pollen grains, dust, bacteria cells, aerosols, etc., as it falls and attaches to the sticky mucus, and it keeps it from going down deeper into the respiratory tract. Here's an example of stratified columnar epithelium. You can see how you have one layer that's more developmental layer, and then you have this consistent next to each other. You can see the nuclei. You can see where the columns are. Okay, there are a couple of things you need to be aware of also when we start talking about connective tissues. Connective tissues consist of various structures, have a variety of materials in them to help them to be part of support structures, etc. They're going to be very commonly having intracellular materials that are like the ground substance, which is made up of proteoglycans, protein fibers, some of them that are going to be involved with the basic capability of handling stretch and pressure, and adding sort of a strength to it. So you're going to have collagen fibers, either as reticular fibers or as a straight collagen longer fibers, as well as elastic fibers made of elastin, which is a different type of protein. You're going to have different types of connective tissue. There is loose, there is dense, and there are some other ones. Let's go through some of them. So the areas of loose connective tissue include areolar, which is a superficial fascia between the skin and the muscle, and you'll find it also around capillaries. Adipose, which is basically, yes, that's the fat that you see, but it's providing an important function for physiologically. It provides for insulation, cushioning, and stored energy. It is also found in the superficial fascia, and it will be found also in yellow bone marrow. Now keep in mind, fat is stored energy in the sense that it can be liberated into fatty acids, put into the blood, and used by many cells for energy. Also reticular, which is more kind of like, think of it as like a really supportive network with lots of spaces in between. And that's what you're going to have with spleen, lymph nodes, thymus, liver, kidneys to a degree. The supportive framework in some cases is basically almost just an array of fibers, etc. Think of the word rete. Rete, the prefix of reticular, really comes down to net or meshwork. This is sort of a generic example of a connective tissue. So what are you going to find? You're going to find elastic fibers, collagen fibers, those big thick fibers compared to the small wimpy looking little elastic fibers, but they all play an important role. You will also find capillaries coming through here with red blood cells that supply nutrients. You will also find the possibility of adipocytes. Notice that the space in the adipocyte, most of it is used to contain lipids. One of the things you find when you do a slice and you see a microscopic slide of adipocytes is that because of the solutions that are basically making fat soluble, these cells look very empty and spacious because they've literally just washed out most of the lipids that are in here. Okay? You'll also find fibroblasts, which help to lay down some of these fibers, as well as you're going to find fixed macrophages. Fixed means that they stay permanently here. Free macrophages come and go. They may visit from the bloodstream and then leave afterwards. Plasma cells are another type of immune functioning cell. All of these are defending against foreign antigens, foreign proteins, bacteria, viruses, fungal cells, etc. Also, from pigmentation, you will have melanocytes, which produce, yes, melanin. You will also have lymphocytes, neutrophils, and eosinophils, as well as mast cells, which also add sort of a protection for this. Now, the mesenchymal cell adds to the productivity of some of the fibers and adds to the ground material. Remember that all this space here that you see that's in purple, that's all ground substance, okay? So let's go through where would we find these. Areolar tissue can be found in the tissue from the pleura, which kind of are the membranes that protect the lungs. Areolar tissue has a very open framework, as you can see here, and all the cells of connective tissue proper are found in the areolar tissue. So you will find macrophages, fibrocytes, adipocytes. Notice that it's empty. It's not because that's a lumen. That's actually just been washed out of all of its fat, fatty acids, lipids, etc. from that cell. But that cell's still there, and you still have a nucleus in there too. Okay, it may not be easy to be seen. You will also find, of course, the elastic fibers and collagen fibers here. Areolar tissue is involved in cushioning organs, providing support, permits independent movement. Phagocytic cells, such as macrophages, etc., provide defense against pathogens. Adipose tissue. Now, here's a solid example of adipose tissue. Lots of adipocytes. Notice this is the nuclei of these fat cells. Notice the rest of it is all empty and washed out because the adipose was removed during the staining process of getting this tissue slide here. But adipose is very important. It's not only a reserve fuel, but it also insulates against heat loss and supports and protects organs. And as you can see here, here's your adipocytes here, and here are the nuclei. So you're going to see them here, and you'll see this here, and here, etc. Moving on, reticular tissue. Now, this is an example I use of the spleen. Here are the reticular fibers here. See these dark lines here? That's from the stain that identifies the fibers, but it's a very loose network. And reticular cells lie on the network. The rest of this is occupied by mast cells, lymphocytes, etc. Because this is a storage organ that also contains a large number of white blood cells, immune cells, and guess what? You're going to find some red blood cells over here too, because the spleen processes old red blood cells and destroys them. Okay? Now, we move from here to dense connective tissue. The types of dense connective tissue are dense regular, dense irregular, and elastic tissue. Now, you might sit there and say, what does that mean? Well, we're going to show you where it's going to be present. Dense regular tissue, think of it this way. The collagen fibers are all parallel, so they're all stacked on one another. So they have the great capability handling stress, stretching, and holding together things. It's almost like you're having a continuous sheet of steel fibers that hold on to things, okay? And so what predominant cell you're going to find there is fibroblasts, which help make these collagen fibers. So you'll see these very commonly in tendons, ligaments, and aponeuroses. That's kind of a sheet-like structure of a tendon. We'll get more into that when we get into the muscular system. The dense irregular connective tissue. This is multi-directional array of collagen fibers. So not just merely just pulling, but it can handle in X, Y, Z, in all these different directions, the tugging and pulling on things. So where would you see these prominently? The dermis of the skin, the capsules around organs, and the coverings for brain, spinal cord, and nerves. Oh yes, and one of the predominant cells there is again fibroblast cells. So to help between regular and irregular is irregular. You're not going to see this nice stacked up arrangement of collagen fibers. Regular, you will. They'll all be sitting parallel. Elastic tissue. The difference here is that instead of having a vast amount of collagen fibers, it has usually a much larger amount of elastic fibers. So it has a great amount of stretching, but recoil. So it's allowing for flexibility. And you'll see this very commonly in elastic ligaments between vertebrae and in the walls of large arteries, which have to expand and contract as blood is rushing through them. Here's an example of connective tissue proper. See the collagen fibers as they move. They are like stacked on top of each other. The nuclei of the fibroblasts are darkly stained right here. So this is very common. You would find this in a tendon, in a ligament, etc. Okay? Now, you do have a few elastin fibers, but mostly the major fiber that's present is collagen, and the major cell type is a fibroblast. In connective tissue, the dense irregular type, here's a fibrous capsule at a joint, okay? And so what's going to happen is they're going to be coming out of the plane and in directions, left, right, all over the place. So the collagen fibers can handle a lot of tensions exerted in many of the different directions, but it provides a lot of structural strength. You're going to find this very commonly in fibrous capsules of organs and joints, the dermis of the skin, keep that in mind, as well as the submucosa of the digestive tract, okay? Notice that the predominant cell here again is fibroblasts. Now, we're going to move into the integumentary system. You might note that I missed, and that wasn't intentional, the muscular system, the nervous system. Okay, so muscular tissue and nervous tissue will be dealt with later on. And actually, we'll come back to the section exercise 5 to deal with those when we get into muscular tissue and nervous tissue down the road. Okay? Don't panic. I do expect you to go over some of this as I have a subsequent later on lab dealing with these type of tissues. But what we're going to do now is focus on the integumentary system, the skin. The primary layers are the epidermis, This is all what makes up what they call the cutaneous membrane. And then we have the dermis, which has two layers, a papillary layer, which is next to the epidermis, and then a reticular layer, which is much lower, which contains a variety of accessory structures, and eventually is at the boundary of the subcutaneous layer, which they also refer to as the hypodermis. You want to keep in mind that there are going to be some accessory structures that are important. Hair shaft. We're going to have the sweat gland, the sebaceous gland, and we're going to have, of course, exit ports for the sweat. This is a pore of the sweat gland duct. We're going to have also down in here nerve fibers as well as blood vessels, artery and vein, and this connection here of a blood supply that is called the subpapillary plexus. Okay, so you see how it just comes through this area? Because eventually we have to have these little capillary beds that pop up on this papillae right here, because they're going to have to diffuse nutrients and take away waste for the germinal layer of the epidermis. Note that there are no real blood vessels in this layer, so it's all by diffusion. Now, before I go, I want to just bring up a couple of other important structures that are accessory, but they're very important. Of course, you've got the hair follicle, you've got the erector pili muscle, which helps the hair stand up, and that also accounts for your goosebumps. But then you've got the sensory structures, such as the lamellar Pacinian corpuscle and the tactile Meissner corpuscle. Okay, so that helps account for the sensory input that you get from your integumentary system. So let's go over the layers again. Epidermis, which is basically made up of stratified squamous keratinized epithelium. Most cells are called keratinocytes. The dermis, which consists of two layers. The papillary layer, which is composed of areolar connective tissue, and that's right up against the beginning layers of the epidermis, and the reticular layer, which is consisting of dense, irregular connective tissue. The hypodermis is below the reticular layer of the dermis, and that's consisting of areolar and adipose tissue. Now, when you deal with different types of skin, thick skin versus thin skin, there's just one big difference you're going to see. Thick skin has 5 layers. And when I'm talking about 5 layers, I'm talking about those layers found in the epidermis. Thin skin is going to only have 4 layers. And again, that's all the layers that are found in the epidermis. So let's talk about these layers of the epidermis. So here we have the epidermis. Notice that you're going to have these unique structures that kind of are sort of like crests and ebbing, or valleys and mountains that you see here on this epidermal ridge, and they coincide at the same time with these dermal papillae, which come to the top here and drop down, and there's a blood supply that's very rich down here in the dermis. The 5 layers that we're talking about are the stratum basale, the stratum spinosum, the stratum granulosum. Now, there's a thin layer that isn't easily seen. It's called the stratum lucidum. We'll talk a little bit about that. And then we have this basically layer of dead cells really here. That's the stratum corneum. Okay. And underneath here, of course, is the papillary layer. See how it comes up, little papillae right there of the dermis. That's thick skin. You will not find in thin skin the stratum lucidum. You may also note that there are some reduced layers here of the stratum corneum for thin skin. You get thick skin, okay, if you have a lot of pressure, a lot of erodive or frictional forces put upon it, and so these layers build up. Now, these are all rich, keratinized, but dead cells. Notice the difference in the thickness of the thick skin for the corneum phase, and then for the epidermal phase here of the thin skin. Big difference. Still a lot of living cells here, but they go through morphological and biochemical changes that change them basically into dead, flattened out cells. Okay. So again, you will look at this in your book and you will review the striatum basilae, the spinosum, the striatum granulosum, striatum lucidum, and the striatum corneum. The corneum is sort of the dead cells. We shed off 5,000 to 50,000 particles an hour. And that includes, of course, hair cells, eyelashes, and skin cells. That's the interesting thing. So when you see doctors scrubbing up and they scrub their arms and everything else, what are they doing? Trying to remove all the looser layers that are dead above them so they don't have one skin flake fall into a patient. Just to help you to think about this for 1 second. Dead cell would have, of course, possibility of bacteria on it, fungal spores, etc. And you have that one skin flake fall off and enter into a person. Perfect growth medium, moist, dark, warm, lots of nutrients around. Bacteria would have a party. So that's why you have to basically gown up and scrub up before you get ready to do surgery. Striatum basically, that is the layer that's also called the striatum genitivium. It's basically the generational layer where you have mitosis very active there to make replacement cells that develop and then go through morphological changes, structural changes that you see them going from basilae to spinosum, spinosum to granulosum. If you have thick skin, you're going to have a few of these thin layers here called the lucidum, which is like about maybe 4 or 5 cells thick, but they're very thin, and they call it lucidum like glass. You can really see through them. And then you have, of course, the corneum, which are basically dead. As you're going from granulosum to corneum, the cells basically break down before they've been building up with lots of protein, and particularly keratin and keratin precursors, but eventually they flip over completely. All the organelles, the nucleus dies down, etc., and these just become sort of cell membranes with lots of protein in them, and some of that protein actually spills out in between the cells. So, as I said to you, where's the striatum lucidum? That would be between the corneum and the granulosum. If you follow through these, the basilae is the site where you're going to have the epidermal layer that's going to have one row of actively mitotic stem cells. And these cells are going to develop and continue to keep building up. You're going to have several layers of keratinocytes when you get into the spinosum. They're all going to be unified by desmosomes. The cells contain thick bundles of intermediate filaments made of pre-keratin. As they develop and continue to move upward here, these keratinocytes are going to start going into being more flattened. Organelles are deteriorating, cytoplasm full of laminated granules, basically will release lipids, and you will also have the production and buildup of this keratin hyaline granules. When you go from here to there, going from the granulosum layer to the stratum corneum, dead cells, 20, 30 cells thick. Now, why I bring this up, because sometimes I will state this. Some people get told all sorts of stuff in our cosmetic and skincare industry, and they say, feed your skin. Well, how can you feed the outermost layers? They're dead. They don't need nutrients. They might need some replacement oils from harsh soaps, etc., but for the most part, you're not feeding anything that's living in these upper layers. The corneum is the most superficial layer. It's anywhere from 20 to 30 layers of dead cells. Thick, it's represented by flat membranous sacs filled with keratin. The glycolipids end up in the extracellular space. Now, by the way, there's a few of the cells you've got to be aware of down here. One is the melanocyte. Melanocyte is going to make granules of melanin. It counts for skin pigmentation. The melanocyte will have some of these granules spill out. They will be in between spaces. As you can see, there's also cytoplasmic extensions of the plasma membrane of the melanocyte. And some of the granules will actually be picked up by some of the other cells. Okay? You're also going to have a sensory nerve ending here, right here. Okay? And that's called the tactile or Merkel cell. Here's on this papilla of the dermis. Notice there's no blood vessels here, as I said earlier. This is fed at this level right here at the striatum basilae level by diffusion from the dermis. That's it. You also have an epidermal dendritic cell. This kind of monitors the immunity here and will act and notify the immune system if there is something like an exposure to a particular antigen or actually a pathogen present. When we get to some of the accessory structures, we won't spend too much time on it, but I want you to get introduced to them now to be prepared for the upcoming lecture parts on the skin. First off, hair follicles. They consist of the hair bulb, which consists of both the hair matrix and the hair papilla. Keep in mind something. By the time you see hair on the top, as it's coming out of the skin, yep, it's dead too. Attached to the follicles are erector pili muscles. These are very small, smooth muscles that respond, and they will respond into sympathetic nervous stimulation, and that accounts for things like goosebumps and pulling the hair up straight, etc. Sebaceous glands produce sebum. This is secreted by holocrine secretion, which in other words, the cells rupture as they produce certain vesicles of oil, but they don't exocytotically release it. The cells will rupture and release cell material as well as the oils that are necessary to lubricate the hair's shaft. Sweat glands, though, are a little bit different. Sweat glands you're going to have are merocrine, otherwise referred to as eccrine. They are distributed throughout the body. Now, in essence, I'm saying to you that there are two types of sweat glands. Merocrine's one, distributed throughout the body. Apocrine are glands restricted to the nipples, armpits, and anogenital region. They start kicking into function about puberty time. Okay? Now, you can see here the integumentary system broken down to basically your cutaneous membrane, basically made up of epidermis and dermis. Your accessory structures, which really are hair follicles, and you can see here in this follicle here, the matrix here that feeds the nutrients, that builds up the cells in this hair shaft as it's being developed. You have your exocrine glands, which include sweat glands and sebaceous glands. And then we finally have the nails, which basically protect and support the tips of fingers and toes, okay? No, this down here is the bone. What you have is actually the nails going right down into here in the nail bed. The reason nails get to be so interesting, in part, is not for beauty and fashion. Actually, you can see a lot of activity from looking at the nail bed. If you just squeeze down very briefly and pull away, you'll see that the white of that area went back to being somewhat pink, regardless of the person's ethnicity or pigmentation or anything else. That's why we use pulse oximeters and we read the blood flow underneath the nail bed, because that's where you're closest to see the actual blood flow through the capillary beds underneath the nail bed, okay? That's where you get the wonderful information such as PO2 and pulse, okay? Two critical clinical factors that you use in determining the health and well-being of a patient, okay? Pulse oximeters used to be with a big massive device, and you wrap this little IR beam through the skin and it would be detected by another type of photoreceptor, etc. And now we've got these little devices that kind of clamp on the tip, and they can electronically, by a Bluetooth, go right over to the doctor's laptop, or you can read the numbers right off of this little device, and it's fantastic. But let's move forward. I want you to review this anatomical model. I want you to review the videos about the skin, because there will be probably some reviews of some of the anatomical structure of the skin, particularly both the cutaneous membrane part. Don't forget also that there's a subcutaneous layer here called the hypodermis. as well as the accessory skin structures. Okay, these are very, very important. Keep mindful of how do we get fingerprint patterns. Well, this is the differentiations that we see between the epidermal ridge and the dermal papillae. And these are raised structures. And here you can see as an example of it on the thick skin. Now, by the way, if you take a real close look here, and this is a scanning electron micrograph, these little pits here, guess what? They're sweat glands. So when you drop a finger down and they do all that fancy forensic work and they're looking for a fingerprint, what the fingerprint is partly telling is an outline pattern of either sweat and or sebum. And that's why they use powders, which will attach to it and give you the image of the real fingerprint. But one more little trick to tell you. It was around the late 1990s, I think it was 99, that Quantico, the FBI Research Center, had been able to develop the technique, are you ready, to extract DNA from a fingerprint for use in forensic identification of somebody. So in other words, what you leave behind is not just merely your fingerprint, but your DNA, and they can now extract it and use it as the identifying information for a suspect. Let's take a little bit closer look at those nails for a minute. This is the direction of the growth. This is called the free edge of the nail body. Underlying blood vessels give the nail its pinkish color. This is the luna here. And what you have is the epinichium right here. This is the edge. We have what we call the proximal nail fold right here. So the epinichium would be considered the cuticle. Okay, here's the nail root bed down here. Of course, you've got the epidermis, dermis, and phalanx. This is basically bone. The hyponychium is right underneath here where the skin comes in contact with almost the free edge of the nail body. Again, why is this so important? Because the information that you can gain from right here is tremendously important. And I will also state this. You can determine certain nutritional and other metabolic activities from nail. You can tell if somebody's under chemo. They're going to have these white, big, prominent spots on the nail. You can tell the development of copper buildup, such as Wilson's disease, because you're going to get kind of a greenish or bluish pigmentation right at the edge of that nail. Okay, so we're just about done here. I just want you to focus on a couple of things. You have a handout that I've provided. You can type up all the information in that handout and upload it. It's got to be uploaded by the Friday of this week. Now, I put this as a difference from the lab activities you'll be doing in the classroom. Even if you were in the classroom now, I would give you the opportunity so that you could download the sheet, fill out all the questions, fill out all the information, fill out all the diagrams, etc. And when you upload it, I've got now a hard copy. Do not ever email me any work because I won't accept it, okay? Part of that is protection because attachments can also contain viruses and trojans and other things to mess up your computer. I will grade it, and you will then be able to see the grade and the reason and the feedback about how you got the 100 or why you didn't get 100, okay? Now that's going to be for exercise 5, exercise 6. You want to complete all those pages. And also, you may get questions as you go through the rest of exercise 6 or part of exercise 5 that you're not going to hand in to me, but you should be filling it out to help you to use those exercises as review for the up-and-coming lab practical exam, okay? I want you to review also all the lab videos for this week. I've got some really nice ones. I've tried to keep them under the 10-minute margin, but the ones for slides, for the histology, for the epithelial, and for the connective tissue are really good. Also, there's an excellent material on the skin. Okay. You want to prepare for next week's assignments as well, because that's going to be involving 5.6, 5.7, and 7.1 to 7.5. We're going to start going into bone, osseous tissue. Well, I hope you have a nice day.